Thank you for that very kind invitation. It's lovely to be here this evening to talk about stress, our favourite topic. I think everyone intuitively understands what stress is. As humans in everyday life, of course, we have to deal with stress. Some cope better than others. And to talk this evening, what I want to do is, I guess it's reasonably high level. It's ideas, it's some data, but also trying to understand how certain regions of the brain help us cope with stress. And in green there, you've got two images there, you can decide which, left or right. In green, in our frontal lobes, we have a region called the prefrontal cortex. It turns out that this region is incredibly important in what we call top-down control. It's experience-led, so in other words, we need to learn to experience stress, and those experiences shape plasticity or learning, and how we deal subsequently with stresses. There are other structures in the brain I'll talk about this evening. One structure there called the amygdala. I'm sure all of you have come across amygdala there, and it's adjacent to the structure called the hippocampus. Those three areas, I guess, will be the key regions of interest in tonight's talk, so keep those in mind. I always like to start historically. And I think this is toggled. No, it's not working, but anyway, we could just do it manually, could we? There we go, good. Yep, so historically, it was Cannon that came up with the idea that stress is essentially a perturbation. It affects a negative feedback system. And classically, in any negative feedback system, you need a way of monitoring a particular variable or signal. That might be something in the blood. In the case of stress, that's the hormone cortisol. You need receptors which pick up that signal. And you need a corrective mechanism that when stress is ongoing and needs to be inhibited, there's a way of shutting it off. But it was Cannon that realized that stress is a threat to homeostasis. Now, homeostasis, of course, we have drive states like hunger and thirst. We get cold, we get hot, we can regulate those bodily sensations. It was unusual at the time to think that stress was also a threat to homeostasis. But it turned out to be an incredibly useful starting point to thinking about a newer concept called allostasis. And I'll talk about allostasis as essentially the wear and the tear on the body. That when we endure stress, there are changes which affect our homeostatic set points. It's called allostasis. And I've mentioned this hormone already called cortisol. Cortisol is a hormone which is secreted from the adrenal glands. This is a pyramid-shaped structure on the top of each of your kidneys. There are two main regions, the outer cortex, the medulla, which releases catecholamines like adrenaline, noradrenaline, and is secreted into the bloodstream. And then the one which is extremely important for stress is a glucocorticoid, which is released from the inner margins of the adrenal gland, and that's called cortisol. I'm sure you've all heard of cortisol, and I guess your first impression of cortisol is it's always doing harmful things. So when cortisol is high, it tends to be viewed in a rather negative way, and when it's low, well, we're controlling stress rather well. Well, there is a phenomena called the cortisol awakening response. So in the first hour when we wake, cortisol secretion is at its highest. It increases, it increases, and increases, and it's thought that it's an ancient adaptive mechanism which anticipates the impending day ahead. So all of the energetic and demands that will be placed on us, we've learnt those in the past, and we know that when we wake up, we're about to meet those challenges, the so-called cortisol awakening response. This has been a myth now in psychology for decades. And there's a new paper that's just come out which has questioned whether it exists at all. And it turns out probably it's a circadian oscillation. And actually, the peak was happening before you wake up. And I'm actually writing an article on that now, dispelling this myth, the so-called cortisol awakening response. But nevertheless, cortisol is entrained to be high just before we wake, and it drops, and it's its lowest levels just before we go to sleep at night. But it is the mediator of our stress responses. When we think about stress, stress has non-uniform impacts on the body. This is a field in psychology which has been studied for decades. In fact, some of the first studies were carried out well over a century ago by Yerkes Dodson, generating these so-called inverted U-shaped functions, that we all need some stress in order to do what we do during the day. There is an optimum, a level of stress intensity, which gets us into this green zone we call eustress. And when we're working this optimal level, this is really where stress is helpful. I guess when we think about everyday effects of stress, we think more in the red zone, distress. Chronic, unmitigated, uncontrolled stress, which is unpredictable and difficult to manage, and there are vulnerabilities in all of us, which means that some of us may be particularly susceptible. You can form or fall into this toxic stress region, the red zone. But to understand stress, you need to understand that stress is non-linear in its effects. Now, we're always asked, and we work on grant panels like the Medical Research Council, to think, well, what is the unmet need? Well, there is an extraordinary unmet need for mental health disorders. We can think of the cost to the government purse of $118 billion. That's a conservative figure around mental health. We can think about stress-related disorders more specifically. So we can think of depression, anxiety, addiction, metabolic and cardiovascular disease. That's a very conservative number at $12 billion. Class A drug abuse, alcohol abuse, probably in the tens of billions in terms of cost. And think of the GP appointments that are generally stress-related. It's an enormous burden and unmet need in society. Now, what we have currently in terms of medication, we have selective serotonin reuptake inhibitors to treat depression. They have anxiolytic activity. These were developed, discovered in the heyday of psychopharmacology. There was a period in the mid-1950s, 1960s, when there are tremendous discoveries around psychoactive compounds. And it is remarkable that in the decades that came next, there's a so-called dark period where nothing really happened. And we're still using SSRIs. They are effective in some people, but about half of people, maybe lowest estimate, about 30% of people, simply don't show efficacy, or these drugs don't show efficacy in these individuals. So-called treatment refractory depression. We're still beginning to understand what those mechanisms look like, but there are some new kids on the block, so-called fast-acting antidepressant drugs based on ketamine, which is an NMDA receptor antagonist, called S-ketamine. It's a nasal spray. Remarkably, unlike the SSRIs, which take 2 or 3 weeks to work, these can work within a single day. So you get benefit the next day after taking this nasal spray. I'm going to talk then more about magic mushrooms, psilocybin, hallucinogens. There's great excitement now that there's an untapped science which we're only now beginning to understand, and remarkably maybe the panacea for thinking about how we might treat stress-related disorders in the future. And I can't give talk on stress without mentioning the pioneer of stress research, which is Hans Dalley, a Hungarian-Canadian endocrinologist. If you just Google this chap, you'll see lots of great quotes. The one that I find as a psychologist the most telling is it's our interpretation and appraisal of stress which matters. So any given stressor that all of us may experience, if it's a common stressor, each of us will react differently because our individual interpretation is different. So it's not stress that kills us, it's our interpretation of the stressor itself. So if we're thinking about stress controllability, we need to understand how we interpret stress. I can't give this talk without saying why we have a stress system at all. the stress systems emanate from the hypothalamus, with the pituitary gland releasing cortisol via the adrenal gland, is intended to be adaptive and helpful. The issue has always been in humans that we overuse this system. So, for example, the adaptive response or survival response for starvation is to conserve energy. In modern societies, conserving energy leads to eating disorders, obesity, and heightened BMIs and so on. If we conserve or if we become to combat dehydration, we might conserve fluids and electrolytes that can lead to hypertension. If we can think about anticipating adversities in the environment, if we do that too much, it leads to anxiety and hypervigilance. So what is adaptive, and what these systems are trying to help us with, quickly become maladaptive if they're overused. And just drawing on biology more generally, I work with mammals, cross-species, cross-scale, to understand stress. But we can learn a lot by looking at plants as well. And as plants also experience stress. And in fact, one of if we think about how we're going to treat stress in the future, we need to understand epigenetics. We need to understand how the environment plays on the genome to control gene expression. And it turns out that plants are stressed, they're sessile. In other words, they show no locomotor activity, they're rooted to the same spot. They have to endure everything that's thrown at them, from high to low temperature, you name it, pests, weeds, they're stressed. And the epigenetic signaling can be passed on to the next generation, to the next generation, next generation, just like humans. They're transgenerational epigenetic signals. And if we look within plants, some of the main players involved in stress responses in plants are mitochondria. Mitochondria, of course, are the energy source. They're involved in biogenesis. But of course, you need that process to enable us to control stress. And if we think of thermotolerance or the memories that we hold for, say, low temperatures, they're all held within the epigenome, in plants as it is in humans. So, back to Dalley, the general adaptation syndrome, or GAS. Essentially, there are three phases to stress. The first is alarm. This is what we're all used to. If we're suddenly confronted with a threatening stimulus, we show an acute change in cardiovascular output, heart rate increases, blood pressure increases. These are all adaptive. It's helping us to avoid a potential threat in the environment. It's very short lasting. The alarm phase is purely an adaptive phase. Now, if stress continues, we develop into a second phase called resistance. Resistance is mediated by cortisol. The alarm is by the catecholamines, noradrenaline, adrenaline. The next phase, resistance, is cortisol-driven. This phase is there to strengthen, to anticipate, and build those adaptive mechanisms which enable us to control stress on longer time scales. However, if stress continues, it's intense, it's uncontrollable, we can get into this phase called exhaustion. And I think some of us will probably know what exhaustion feels like, sleep deprivation, hypervigilance, worry, anxiety. If this continues, it builds this process called allostasis. The stress gets under the skin, it accumulates, and this concept of stress memories, I think, is a potent one. To think that stress can be remembered, it's persistent, and it can accumulate and show plasticity. And getting back to this idea of epigenetics, likely that is the source of these so-called stress memories. And when humans are confronted with stress, they show coping reactions. Now, coping reactions are broadly defined reactions to stress. We can think in the stressful event in green there, leading to our individual appraisal of whether or not we're able to master that stressor. There can be psychological outcomes leading to emotional changes, anxiety, cognitive impairment or changes, and there can be physiological things we can measure: autonomic output, heart rate, pupil diameter, skin galvanic responses. All these are very measurable coping responses. The issue with stress is that coping is there to mitigate the initial stressor, but humans will generate secondary coping responses, which, if they become the dominant way of controlling behavior, they themselves become the stressor. So, for example, if you have a busy lifestyle, you're stressed at work, you have tight deadlines, you might find comfort in drinking alcohol as a way of alleviating, displacing stress. Now, of course, you can overuse that coping mechanism and the alcohol becomes the secondary stressor. It's a very well-known pathway into some of the substance use disorders. The primary stressor is being displaced in some way by a secondary stressor. Now, humans can control stress, and as mentioned, this green area, the prefrontal cortex, is experience-driven. We learn about our experience of stress, and we use that experience to suppress, through a top-down mechanism. This is the front of the brain here. We're taking these top-down inhibitory mechanisms over other regions of the brain, including the amygdala and the striatum. The striatum is an area involved in habit-based learning. One thing about stress, if we're exposed to too much stress, we tend to operate in a habitual way. We tend to act in a very robotic, automatic way, and that's controlled by one area of the striatum. If the prefrontal cortex becomes compromised, there's too much stress, it's weakened by stress, the striato-amygdala systems begin to dominate for control over behavior. This is where we're beginning to tip into the so-called toxic phase of stress. So we want to stay in the green zone, resistance. We need the prefrontal cortex, the networks that support the prefrontal cortex, to be integrated to allow top-down inhibitory control of the amygdala and striatum. And this is a curious phenomena of stress, that people who are exposed to stress, who manage poorly with stress and show allostasis, i.e. changing morphology, architecture, cellular makeup in the brain, it's the amygdala which shows a growth process. You can look at neurons. This is a depiction of a neuron within the amygdala there. We can look at acute stress. We can look at more chronic, lingering or repeated stress. As a growth process called spinogenesis. A spine is where another neuron is making a synapse onto the neuron, and for repeated stress, we see this increase in the number of synaptic connections with the amygdala, this hypertrophy, if you like. And we know that if you have a brain scan and people who are very stressed, if you've got post-traumatic stress disorder, or you've been in highly traumatic situations, that's the amygdala will show enlargement. So volumetric enlargement is one of the outcomes of repeated unmitigated stress. But curiously, if you look at other brain regions, for example, that green prefrontal cortex or the hippocampus, some of you will know hippocampus is a structure involved for declarative memories, episodic memory. Those regions show the opposite changes. They show atrophy. So there are reduced synapses. If you look at the complexity of neurons within prefrontal cortex or the amygdala and look at the density of spines, they begin to shrink. It becomes less organized. The dendritic trees, which collect information from the neurons, begins to shrink as a result of chronic stress. So here we have, again, this non-uniform impact of stress. The amygdala showing growth, prefrontal cortex and amygdala showing shrinkage. Now, let's get back to the types of stress that humans face on an everyday basis. The stress systems, if we think of survival pressures, were intimately linked with the immune systems because it made sense that the predators at large and there were aggressive dyadic encounters, that it would make sense to be in the alarm phase, to be vigilant, have a cardiovascular performance that was ready and primed, an immune system which would be suppressing inflammatory responses. So evolutionary, it makes sense that those systems are linked. The issue that we all face now is we live in modern, safe, hygienic environments. And the types of stress we suffer and endure are psychosocial. It's the comment from a colleague, or in this case, it's a workplace situation where a superior may be belittling or somehow informing this poor chap here that what they've done is incorrect. It's being listened to by colleagues, and it's a very belittling and very difficult experience. And that chap may go home and incubate what happened the day before. The rumination of what happened then becomes the problem. Okay? Humans have a way of ruminating about events. So the event doesn't just start and stop, right? You might have read this book called Why Zebras Don't Get Ulcers. Ulcers are a consequence of chronic uncontrolled stress. Zebras don't anticipate stress in the same way that humans do. We have this prefrontal cortex. We always plan and consider what's going to happen to us. Zebras live in a terrifying environment, but the stressor happens very quickly. They're conspecific, as be frank, eaten or whatever, and it all goes away. And the stress response goes up, and the stress response comes down. For humans, it lingers, and we ruminate about what's happening. Workplace environment like this, psychosocial stresses, are very difficult to overcome. The other type of stress is workplace stress, occupational stress, civil servants, working in air traffic control, where vigilance is the premium. One small mistake can have dramatic consequences. And we know that people who do this job very well have incredible ways of coping with stress, with switching off their stress response after the end of each shift. Another type of stress is one which can be very high in terms of intensity. It can be completely unpredictable. One event, and it can lead to conditions like post-traumatic stress disorder. We know now a lot about how that happens in terms of how that memory is consolidated. It gets consolidated in the wrong place in the brain. So what we remember is the emotional trauma. We often forget about what actually happened. We lose that bit of the memory. But it tells us that even just one event, very high intense stress, can have dramatic and persistent effects. The other type of stress that we should care about is stress which occurs to us early in life. These so-called adverse childhood experiences, these ACEs, maltreatment, deprivation, neglect. There's all sorts of reasons and pressures in some individuals. And what we know about from stress research is that the developing nervous system is especially vulnerable to stress. The window for plasticity in stress-related memories is the highest when you're very young. And that plasticity is generated through epigenetics, how the environment plays on the developing nervous system. The question I'm going to pose in this talk is whether early life stress is somehow immunizing, building inoculation, and how we cope with future stress, or as some people believe, this early toxic stress, which is bereavement is uncontrollable and has lingering effects, whether that leads to increased vulnerability later in life. There's an enormous amount of work on so-called early life neglect and adversity. They're strong predictors for later mental health outcomes. And it's not just infancy, post-infancy stress, it's in utero. So across the perinatal period, cortisol, which is being released from mum, it's very lipophilic, it crosses the placenta very readily, it can entrain the circadian cortisol cycle of the developing fetus. And we know that with chronic stress, particularly in late first trimester, that can change the trajectory of developing neurons within the central nervous system. And as a very extreme example, the the case of orphans in Romania. They were living in extremely deprived conditions with no care. There was all sorts of issues in those orphans. And there was a remarkable brain imaging study carried out by Edmund Sanuga-Bark in London using MRI, magnetic resonance imaging, and looking at the volume of the orphan. And one of the standout findings was that the amygdala showed an increased volume. And that was bilateral, both hemispheres in these orphans. It's a remarkable study, but telling us that very intense stress early in life can have remarkably persistent effects on these brain structures. And then this notion about early life stress, one theory that I believe in passionately is this called the stress diathesis model, that stress accumulates. We keep the toll, we keep the record, one by one, but we have, some of us, an inherent predisposition for stress, and others are resilient in some way. And if we look at this figure here, you can look at the probability of a stress-related disorder occurring based on your predisposition along this axis from low to high and the amount of stress you actually experience. If you have very low predisposition, it stands to reason, no matter how much stress you endure, you're not going to transfer into a stress-related disorder. However, if your predisposition is very high, even low to moderate amounts of stress, the disorder can manifest, the so-called stress diathesis model. And of course, there are important genes which we know about, which control that. It's also how they play with the environment at large. And there is now a great number of imaging studies looking at how the brain develops, how it connects, and how it responds to early and later life stress. Some people think that early life stress is helpful, some think it's less helpful. The experimental approach, or one of the experimental approaches we use, it's a very mild procedure early in life. Now these are rats. Normally the dam or the mum would give birth to a litter of say 8 to 10 pups, and during the first few days of life, say postnatal day 2 to 5, and then right up to adolescence. By the way, adolescence in a rat does happen. It's about the 3 weeks or so. If you simply take the litter and just take them away into a different space, just for a few hours, and they're all together, and then take them back to the dam, and just do that on repeated times every day. You just do that, and it's very mild. It's disruptive. So it's changing the way in which the dam then interacts with the litter. It's called the repeated maternal separation procedure. And it's just a mild, social, disruptive procedure. And, well, okay, that's interesting. So how does that then affect what those sane animals, how they behave later in life? And there's already clues that maybe there are some interesting things that can happen. Mike Meany has looked at mums, which are generally basically bad mums. They don't look after their offspring very well, they don't lick them, they don't groom them. And this gene here, this is the nuclear receptor. This is the receptor which responds to the stress hormone cortisol. Okay? And in the promoter region, if we're enduring too much stress from these offspring there because they're not getting the maternal care they need, this promoter region becomes methylated. These chemical methyl groups bind to the promoter region, and it stops these trophic factors, it's a nerve growth factor, from binding to promote expression of the receptor. Now what we need is lots of receptor, so when it binds the cortisol through a negative inhibitory feedback system, it shuts down the stress response. We want that. What happens in this case, no nerve growth factor, and basically there's low numbers of receptor, that's the low glucocorticoid receptor expression. That leads to high anxiety in the offspring and high stress reactivity. What happens then is these offspring, because of that maternal care, they remember this, and through this transgenerational epigenetic signal, they themselves become poor mums. And the way they nurture their offspring is also not optimal, and it passes on this chemical signal through the initial stress response. This mum, by contrast, there is a happy ending to the story. This mum is a high-licking, high-grooming mum, well-nurtured, strong affiliation with the offspring. Now, no methylation of the promoter region, nerve growth factor can bind, healthy expressions of the receptor, and the stress response then turns down, and we have low levels of anxiety. Okay? Now, you can see early on, you might have an environment low in resource, low in food, high levels of predation and population aggression. And it can lead to this type of behavior in parents and the way they respond to their offspring. So it can all be environmentally driven, some of these changes. All right, so one of the labs that my group work at is at West Cambridge. We have a MRI scanner. It's a translational neuroimaging centre. We have a 9.4 Tesla scanner. It's high field. Some of you know if you've had scans at hospital, 3 Tesla is about the average strength. So it's a high field. It gives us very high resolution of the brain. There's a few stakeholders there you may or may not recognise. This building's been open for about 6 years now, delayed through COVID, but now we're fully operational and able to do very high resolution longitudinal imaging to understand stress impacts. And one of our discoveries is that animals which have been exposed to early repeated maternal separation stress, if we look at how the brain of those animals as adults is then organised and connected, you can see actually the connection strength between the front of the brain, the prefrontal cortex, shown there in these coloured sort of overlain images, with the amygdala is actually reduced. So all that early life separation has been carried right through adulthood, and now the functional connectivity between the prefrontal cortex and the amygdala is suppressed. And if you look at humans with stress-related disorders like depression, you see exactly the same profile. Reduced connectivity between the top-down prefrontal cortex over the amygdala. So you can see already that there is already these individuals are being compromised in how they exert top-down control over stress. The work we're doing is to understand ambiguity. Ambiguity, uncertainty in our world is one of the most important drivers for how we generate stress responses. Anxiety essentially is driven through our interpretation of uncertainty. We can increase our knowledge of the world by being exposed to it and updating our beliefs. But for some individuals, their belief system is quite rigid and difficult to change based on new evidence and new information. And they inherently have a distrust of new information and become, in this way, intolerant of uncertainty. And this is a new way of thinking about the drivers for anxiety and stress-related disorders. We have a Wellcome Trust grant. It's a large strategic grant with a number of people involved, working at the MRC CBU, our lab here in psychology, members in psychiatry as well. And it's really looking with high-resolution imaging, computational modelling, to try and understand and segregate, based on individual differences, responses to uncertainty. And there's lots of ways you can impose uncertainty on someone. We have touchscreen tasks, for example, where an animal will have to learn that this green stimulus is correct on 80% of time, and it will get a nice food reward. And if it goes to this side, that food reward will be delivered on 20% of occasions. So essentially, that's the incorrect side. Right? And if you're tracking the optimal reward densities, you're always going to try and touch the screen where the CS plus is being presented. And an animal will track that contingency. You can get very good at it. What you can do is you can switch the contingency. So now the side that was incorrect is now correct. And of course, you're going to make mistakes. You perseverate on the previously rewarded stimulus. This kind of rigid behavior is something that's very easily captured and measured. This is a way of increasing uncertainty. This uncertainty, you can change the probabilities. If it's 100%, it's certain this will always be rewarded, and this will never be rewarded. Or you can make it 50-50. It's pure chance, which side is which. You can maximize uncertainty. But there's another concept which is important in how we think about this so-called uncertainty. It's called volatility. I don't know if anyone's come across this term, volatility. And the examples of volatility is, well, when you change a fixed probability, the uncertainty is expected. We know when you go into a particular environment that there's going to be some uncertainty, and you can basically know what the baseline average or probability is. Volatility is when it's unexpected uncertainty. Everything is changing very quickly. So there's an example, famous example here of a cat that could scratch you if you touch it or pat it on its head. Okay? Now there's a fixed probability of just 0.1 that's going to do that on any particular occasion. But sometimes when the mood of the cat takes it, the probability of scratching is actually very high, about 80%. And it fluctuates, and it's very hard to predict. It's very unexpected. Now there's two cases here. If you're someone that's intolerant of uncertainty, right, you don't like these volatile worlds. And if you're a slow learner, then you're going to get scratched more often because your learning rate is not fast enough to update your beliefs. Okay? If you're a very high learner, in other words, when you're in an uncertain environment, you have to learn faster. It's called meta-learning. And that acceleration helps us, right? So this trade-off between how fast you learn in volatile environments can be a determinant of anxiety responses. And we're building some really neat technology, by the way. Some of you might have heard a grab sensor. You can, in the brain now, every neuron expresses receptors. In the old days, we would measure a neurotransmitter like dopamine, noradrenaline, using sensors. It took 20 minutes to collect the transmitter. These genetically inspired sensors, now we can transfect a green fluorescent protein. When the endogenous transmitter binds this G protein-coupled receptor, it generates a fluorescent signal, and we can pick up transmitter release in sub-second timescales. So all this technology now is being developed in Cambridge, and noradrenaline, which I've told you is released into the bloodstream when we're stressed, it's also released in the brain, and it's one of the first detecting systems for stress responses. And it also detects volatility. So when volatility is very high, it's noradrenaline released to the brain is also very high. All right, I realize time is pressing, so I'm just going to get to what I think it's hopefully get us right to the point here. Just some early findings. Ethan was a Gates scholar in my group. He used imaging and found that maternal separation stress enlarges the amygdala. So again, consistent with what I've told you already. We're working on immunological events. So a stress memory, the idea and the general thinking is it could be held in immune cells, in monocytes. I've told you stress and immune systems are tightly connected. And the curious fact is that when we are chronically stressed, we develop a process called cortisol resistance. So these monocytes, these inflammatory, normally helpful cells, change character. And they can traffic into the brain, release chemicals called cytokines, and cause damage. So there's a really interesting mechanism around immunology as well. And we've made some quite remarkable discoveries, Mirel and Linnell in psychiatry, understanding how stress can be carried through these inflammatory cells within the blood itself. And maternal separation does all sorts of interesting things to neutrophils and beta macrophages and so on. Anyway, I'm going to cut to what I think are the next generation of stress research here. And it came about, I guess it was a bit of a fishing exercise. Sometimes in science, you do things which are hypothesis-led. You have an idea, and you have a working hypothesis, you update it with new evidence. This technique called bulk RNA sequencing, you simply take a bit of tissue and you sequence the entire genome and you see what falls out. And we're interested in early life stress and this maternal separation procedure. We're interested in the prefrontal cortex. And in the rat, we used this technique of bulk RNA sequencing. We're a bit agnostic about what cell. We didn't really care. We just wanted to know, are there any genes which are differentially expressed in maternally separated animals? What came out from this analysis was a whole cluster of genes which you think, wow, okay, that's interesting. There were no differentially expressed genes, but they're all within the same biochemical cascade. And it was all to do with mitochondrial signaling. So on the electron transport chain, how we make ATP, all of the genes which were upregulated in male and female rats that have been stressed were all different. One pathway. And then we became really interested then that maybe the stress memories that we hold through life are actually sitting within mitochondria, within the prefrontal cortex. And like all things in science, there's always competitors out there. Carmen Sandy, she's a wonderful scientist working in Switzerland. They'd already cottoned on to the idea that mitochondrial perhaps are really the repository of some of these long-term stress memories that we hold. And I've been in Cambridge now 28 years, and in the Department of Psychology, we're right next door to the Department of PDN. The small door kind of separates the two departments. We never open that door, and we never really talk to the person on the other side. And it turns out the person I really needed to talk to was Andrew Murray. And Andrew Murray had been working on altitude sickness, how mitochondrial change when you go high and low, looking at resilience and so on. And he had a postdoc called Lorenz Holzner was working in his group. And we thought, well, look, if you've got RNA sequencing data showing that there are changes in mitochondria, you need to do a dynamic functional assay, respirometry, which looks at oxygen consumption. Does that change in these early stressed animals? And lo and behold, what we discovered was something quite remarkable. I just want you to focus on this bar here. This is an animal which has been maternally separated in the first 3 weeks of life. It's a very subtle separation procedure. And we've done this assay 9 months later. Now, to a rat, that's basically almost its life at the end of life. So it's really sort of well through middle age. And there is a signal there that we can pick up already. That signal is increased biogenesis. There is improved mitochondrial function within the prefrontal cortex of these early stressed animals, and it's been carried right through their lifespan. But look what happens when you subject those animals to a further uncontrollable stress. It uncouples. And so what looked like early resilience now becomes vulnerability. And so the theory we're working with here is that we can endure a mild stress and we can increase our biogenesis through mitochondrial signaling to improve top-down cognitive control. If, however, we're subjected to further stress, which may be uncontrollable, and we've had an early life stressor as well, it can have super additive effects to produce a second cascade, which means the product of this uncoupling can go through the reactive oxidative species pathway. And the thinking here is that it's this pathway which increases our liability for stress-related disorders. And the next question we had was, well, that was 9 months. What happens? Can we see a signal which is already beginning to start at 3 weeks, just in that early separation phase? And the fact there is already a signal there in our maternally separated animals. It's just now it's sexually dimorphic. It's happening in the males and not the females. So what does this mean? Well, the theory that we have here is I'm going to coming back to this inverted U-shaped function. I've told you that stress has non-linear impacts. Well, the way I like to conceptualize this now is that we can think of stress across the lifespan, and we can think that low to moderate stress actually is what we understand from stress. It helps us. It builds resilience. It inoculates us against stress. There is a plateau which is reached, and if further stress happens, it can tip us into this descending arm of this inverted U-shaped function to increase our susceptibility. So what are the implications of this research for how we might think about new therapeutic strategies to manage stress-related disorders? Well, it turns out that one receptor, which is the gatekeeper to mitochondrial function, is this receptor called the 5-HT2A receptor. I told you about selective serotonin reuptake inhibitors. They block the uptake of 5-HT in the brain. Well, they also indirectly activate this receptor, which is the gatekeeper to how mitochondrial signaling actually carries on within a cell. So it can either increase biogenesis here and increase oxidative capacity, or it can tip into this disruptive pathway and increasing reactive oxidative species in the brain. So it's that 5-HT2A receptor, which is extremely important for how we think about mitochondrial function. And it turns out that magic mushrooms, psilocybin, which is the active compound of psilocybin, activates the 5-HT2A receptor, like LSD and other hallucinogenic drugs. And I'm sure some of you may be following the science. Robin Carhart-Harris worked with David Nutt at Imperial College. My first postdoc, by the way, was with David Nutt at Bristol University. Some of you may know he was the former advisor to the government on how we might schedule Class A drugs based on harms and so on. Robin had a number of studies looking at the effects of psilocybin in humans and how they may be used as fast-acting antidepressant compounds. And I guess one of the summaries that they've come up with is an idea around entropy. The idea that we can look at each of these, this is a depiction, it's a really kind of low-dimensional space depiction of human brain and how it connects. And we can think of each of these circles as communities of neurons within the brain which are connected and are functionally kind of similar in what they're doing. Okay? Someone that's had psilocybin, you can see that both the within and the between community connectivity is vastly increased after psilocybin. Individuals on psilocybin will report increased vividness of their subjective experience. Green seems greener, purple seems more purple, pink seems pinker. How does this get us back to this point of how they may be beneficial in stress-related disorders? Well, the idea we're thinking of is that maybe it is through the mitochondrial pathways, which has been previously unrecognized. Robin and David Nutt's view is that the cortex, right across the cerebral cortex, it's rich in these 5-HT2A receptors. When you activate them, it's like a reset. It changes our beliefs and it allows the opportunity for new learning to take place, increased plasticity. Their thinking is that by doing that within the frontal regions, which is exerting top-down control, it increases the opportunity for changing how we think and process uncertainty and update beliefs more fluidly and dynamically. So there's a few of us, I think, with this idea that we need to strengthen the networks of the prefrontal cortex to exert greater control so that the amygdala administration doesn't gain control over our behavioral responses. So our working hypothesis is that these psychedelic drugs are actually hijacking the biogenical pathways mediated by mitochondria in the prefrontal cortex. So maybe we can give you an update soon on that progress. But there's a number of people I need to thank for all of this work. I won't go through each of those individuals. We're funded by the MRC, the Wellcome Trust, drug companies like GSK and Boehringer Ingelheim. So there I'll stop. Thank you very much.